Okay, so Uri already said the important thing. It's amazing to see the same ideas. And you're going to see the same ideas. We had that in Copenhagen at ARDD last year. At the physics workshop we had there was a lot of the same ideas in different iterations, and I think this will be the same thing. So this comes out of a lot of discussions with Peter. And so I just reiterate the idea he just presented, where there is some sort of phase space, some sort of eigen direction along which stability is lost. So your potential is dropping. Oops, I think I had the wrong button. This potential is lost, and the height of it goes down. And so you're losing stability along that axis, and the respective eigenvalue changes sign, and you get instability. And so then you're more likely to fall out of this well as you lose stability, and that depends on the temperature. And my original talk was about temperature, but I decided to tell you about something else because at ARDD something interesting happened, and so I changed my mind a little bit about what I wanted to talk about here. So this is this idea. So fundamentally you have a bifurcation where you have two fixed points, a stable and unstable one, and they collide and annihilate each other until you have just instability. And at this point, you have no more stable system, you have no more height of the potential changing, and that's the thing that's driven by entropy. So the loss of this potential, this bifurcation in this way of thinking is driven by the entropic effect, which can be out of all sorts of molecules and work on different layers and different layers of abstraction. But at this point, you've lost stability. And that gives you this loss over increasing the probability of transition, gives you the exponential mortality increase that you get from Gompertz law. Okay, so that was what we just heard. Now, the interesting thing is I work with another model organism, not flies, I work with C. elegans, and they're like tiny. If you don't know them, they're soil dwelling, they live in soil, they're about 1 millimeter long, 100 microns wide, so they're like a tiny eyelash. They are 99% self-fertilizing hermaphrodites. And they lay 300 eggs per adult in about 5 days after reaching maturity. And we know a lot about them. I'm just like this picture, so I'm just showing off. We took this in the lab. This is the neurons in the head of a C. elegans, and we had some neurodegeneration models. So they're really cool models for all sorts of diseases, and they're also very widely used in aging and have made some big contributions to the field. And this is what aging looks like in C. elegans. So on top there, you have the guys on day 6. They're happily crawling around, and then on day 16, they're sort of very tired looking and don't move a lot. Some of them are partially immobilized. And then by day 23, they may be twitching a little if you hit them with the platinum wire, but otherwise they don't do much, and then they're about to die. And so this is the life cycle of these guys. And why they're used in aging widely is because they're very short-lived. That's very convenient, right? So they live about 17 days median lifespan, depends a bit on the lab. We have a lot of discussion what would be the correct lifespan for them in the lab, depends on the conditions. But fundamentally, that's what it looks like, 100% alive at day 0, and then all dead by day 30. Now, oh, this is horrible, so apologize. This gives you a very foggy idea about what I'm talking about. But the bottom line is there's a huge amount of literature that says actually they don't follow Gompertz law. So if you look carefully at the lifespans of C. elegans, and I'm very guilty of glossing over this and saying, oh yeah, Gompertz law is universal and we're researching this in C. elegans. Actually, there is a deficit of late life mortality. So they're basically plateauing. So sometime later in their life, they do not show a continued exponential increase in mortality. And there's a discussion whether that's true for humans at extreme old age, but this happens at relatively a lot less mortality rate doubling time. So this is something that is weird enough that people have repeatedly every few years discovered this and talked about it and solved it different ways mathematically by saying, we have to have a biphasic model or Weibull distribution or something, but fundamentally it doesn't quite look Gompertzian. Okay, so what's wrong with worms, right? Why would that be? Why would the worms have a weird sort of curve? Well, one thing about it is I told you they live 17 days, but actually in the wild, they live about a day and a half. So they have about a 50% mortality a day, give or take, in soil. So they live incredibly brutal lives. And all of that is extrinsic mortality. This is being eaten by something. They have this incredibly fast life cycle where they go from an egg to an egg-laying machine in 3 days. And then they burn out 5 days later. So they're essentially just like biological copy machines, right? You throw one egg on a plate, and you get an egg-laying machine 3 days later, and then 8 days later you have 300 copies. So they have a very easy strategy which tells you mortality has to be really high extrinsic, because otherwise the whole world would be made out of worms, right? They make 300 copies in 8 days. It's exponentially, a paperclip catastrophe. So, and this is what the egg-laying histogram looks like. This is work in the lab. We work on reproductive longevity in C. elegans and humans and mice. But you can see like they have this massive peak at day 5, and by day 8 they're essentially done with egg-laying, which tallies with the fact that in the wild by that time they'll be dead. Right? So that means they have a very disposable strategy for living. They have to have. And I spent about 10 years of my life unsuccessfully proving that damage controls lifespan using C. elegans. So I measured all sorts of damage using all sorts of methods for many years and tried antioxidants. I'm going to summarize 10 years of my life in one slide, which is a slide that points out failure, so it's great. Not quite, but okay. So what you have here is the amount of DNA damage in mitochondria and worms as a function of time. And as you can see, between day 3 and day 15, there is a significant, statistically significant amount of damage that accumulates. And for me, that was a great success because I had a p-value for the statement mitochondrial DNA damage goes up with aging, which if you work in free radicals was important. But then we did lots of experiments, and I'll just show you one. This is an experiment where we irradiated worms with either gamma or UV radiation, and we cause a statistically significant increase in the amount of damage. So we give them extra damage. And actually, we measured them later, and they never repair this damage, so the damage stays high. Yet, this intervention slightly extended lifespan in this experiment, didn't shorten lifespan. And you can fry worms to levels that would kill a human, and they become sluggish, but they never die quickly. So you need sterilizing amounts of radiation to kill them. So, and you can say maybe oxidative DNA damage caused by gamma isn't what causes aging. That's fine, but you're throwing a huge amount of entropy into them, right? You're like UV zapping them. It does everything, cross-linking, oxidative damage, DNA damage, all sorts of stuff. They don't care. I mean, they care, but they don't die quickly. Okay, so one way to explain this, and that comes out of discussions with Peter, is to say, well, all of that paradigm you're having here is about this damage here. It's about this idea that there is entropic damage lowering the potential, and that causes failure. But maybe what you're having is an unstable animal to start with. Maybe they're not unstable when they're born, but maybe they're unstable when they become sexually mature. So they essentially just give up on maintaining the soma and just make as many eggs as possible, which makes sense as a strategy. And there's evidence that they even digest their own gut material, like the cells in the gut lining, to get more energy, because it's more efficient than keep it up and function. So they are sort of having an almost salmon-like reproductive strategy. Okay, so if that's the case, then we're really looking at this case for these guys. They are unstable to start with. They have no more barrier. There is no more returning force. And if that's true, you can go back to the equation Peter showed, the Langevin equation, and you can say, okay, so what would happen in an animal like this? And that's the neat thing. Doing a model organism, you can do hypothesis testing. Right? You can say, is that actually plausible? Okay, so here you have 3 sort of zones. Initially, you have like a very flat potential, and at that point, nothing matters. It's just essentially diffusion, right? So there's just stochastic noise. There's no force acting. So you basically just get a Gaussian slowly relaxing. Right? And it relaxes to this barrier here, which is the point at which the exponential force, in other words, the force that pulls you down this slope here, starts mattering. And so that is what sets up the original increase in mortality. So it's just basically the Gaussian sort of slowly drifting apart. Then you get to the sort of part of the curve where there's some actual noticeable force, and you get amplification, exponential amplification down this direction. By the way, this is some sort of state variable, right? This could be in your RNA-seq, it could be the first PC of your transcriptome, or it could be the first PC of your lipidome. It doesn't matter. It is a direction of change that occurs during aging. And you get an exponential amplification there because you have this force pulling you down, because they're unstable. And then eventually you get to sort of hyperbolic phase where you have a quadratic differential equation. And there are lots of jokes we could make here, but fundamentally that's death, right? So you're essentially reaching infinity at infinite time, as Peter said. So that's a bad sign. Okay, so that's the sort of model. And so you have these 3 phases. This is what the trajectory would look like. Early on, the Z coordinate here just drifts around, can go up and down. And rejuvenation is completely possible by random chance. You can be pushed down by just random fluctuations. Then you sort of tip across the boundary. It's not a sharp boundary, but you get into the phase where the forces matter, and it pulls you up, and then eventually you get into the hyperbolic phase and you fall apart. Okay, so that's what that looks like. When you do the first little bit, this is a simulation, just picking some numbers. To make this work, you could interpret them, but it's illustrative, essentially. And what you're looking at here, the animals start on the left here, and then over time evolve, and they're just drifting apart. So they're basically just changing stochastically with time, and there's nothing happening. Then some of them cross the boundary where the forces matter and get amplified, and then eventually some of them cross the part where you get acceleration and hyperbolic disintegration. Okay, and that tells you that the lifespan should be about 1 over alpha. That tells you that 0.1 there is on the order of 10 days. So that, for physicists, that's usually good enough. You're in the right order of magnitude. And then you can run a little simulation. And doing this, so let me just hope this works. I was meaning to accelerate this, but I don't have a mouse, so I can't do that. So this is a population of these guys just doing their thing. And, running, just randomly drifting. There's a reflecting surface at the bottom to make that a little easier. And then some of them will dip through the part of the curve where the exponential forces matter and then cannot drift back. You can't get back from there, but it just increasingly accelerates up and then eventually get through the hyperbolic boundary and essentially get it. That's the sort of terminal disease that immediately kills you, okay? So that's great. That's what the simulation like this looks like. I'm not going to let this run to the end because it takes too long. But one thing that you will see is if you run a lot of these simulations, that it's completely possible to just drift forever. If you're lucky, and the guy down here, the whatever green guy, will just go all the way to the end and just at the very end go through. So what this means is that unlike the things that we talked earlier where there's an entropy increase degrading your potential, if that's what's driving it, then at the order of this time span, there's actually not a hard upper limit for lifespan. You can just stay outside of the acceleration phase for long periods of time by just stochastic drift, okay? So we make the population bigger, you will always find an animal that lives longer. And if you do this, you get a lifespan out of this, you count all the things when they cross the barrier, you essentially get something that looks very much like a lifespan curve. You can calculate that too. It's an Earth function. So you get a realistic looking lifespan and you get a mortality plateau at old age. So it sort of reproduces some of the features. So that's all it is. It's a toy model that you can think about and that reproduces some of the key features. And I like toy models because they make you think, I think, more cleanly. So that's the experimental data and that's the little simulated data. Now, the thing about this is, so a stable species, you would expect a Gompertz mortality without plateaus. You always get an increase until mortality basically becomes 1. You expect a hard maximum lifespan because you can't really outrun an exponential failure. And there's no memory of past perturbations. If you wobble this, if you do something on this axis, if you do something to Z, it will forget as long as you don't kick it out of the barrier because that's the whole point of a homeostatic system. Whereas in an unstable animal, like we think C. elegans is at least post-reproductively, or after starting reproduction, you get late life mortality plateau. You will get an absolute you will not get an absolute maximum lifespan. You make a large enough cohort, there will be some crazy animals that live longer than you expected, and there's there's memory of past perturbations. You can push them on the Z and they will remember that that happened forever, as Peter pointed out. Now, at this point, I was outside ARDD having a cup of coffee, and Colin came to me, and he said, look, I have this weird worm data, I want to explain it. And the cool thing that I like about toy models is sometimes they can help you explain something you can't otherwise explain. I'm going to keep this very short. These slides are stolen from Colin. So this is the DAF2 mutation, famous aging mutation in C. elegans. It's the first single gene mutation, or one of the first single gene mutations that extend lifespan in any organism dramatically in '93. And Cynthia Kenyon, who discovered this, had done an experiment where she did RNA at different ages. So she went and did this intervention at different ages, and she showed that the effect gets smaller. And this is what you expect from damage accumulation theories of aging, right? You're saying, I'm doing this thing, it reduces the amount of damage accumulation, and if I do it later, more damage has already accumulated, so the effect diminishes. Okay, so this is seen as, or I used this for years, as evidence for damage accumulation driving aging in C. elegans, because it means there's essentially the past memory of this, or the past record of this damage is there, and if you wait half their life, half the damage is already done, and you can't undo it, but you can just reduce future damage. Now, if you now go and you look at Colin's experiment, he did it just slightly differently. Instead of using RNAi, which it turns out the worms can't eat very well when they're old, therefore the efficiency of the intervention drops. He did this with an auxin, essentially a chemical trigger, a hormone trigger, and he did this at different times late in life. So he basically switches off DAF2 at day 20. And he gets this weird result that suddenly the animals that get switched at day 20, when already 80% of them are dead, suddenly switch to the mortality of the animals that have always been treated, and therefore survive, some of them survive as long as the ones that have always been treated. This should not happen in the damage accumulation theory unless you build some sort of rejuvenation loop into here. You're basically saying, oh, this removes a lot of damage. And Colin and many people have worked on this and trying to look at what kind of damage might be removed, what's actually happening, and so on and so forth. But because we were discussing models, of non-aging animals, I was basically wondering whether this comes for free out of this little model and way of thinking about non-aging animals. So what happens when you do this? Look, so the lifespan is twice, and therefore, because I told you it's 1 over alpha, all you have to do is essentially just half the alpha parameter. For visibility, I also changed G, but just because you want to have 2 lines. And so basically you get just this one modification and you run the same simulation. And what you get is you get pretty good estimate of what the DAF2 lifespan looks like. So in other words, you just half the alpha and you get the right lifespan. So that is nice, it's robust towards this. And then you run a simulation where you do the switch. So this is a stochastic simulation where you run for the first 20 days at the wild type mode, and then you just switch the barriers, alpha and G basically, to what DAF2 would be, and you ask what happens. Okay, so if you do this, you first at the beginning get exactly the same you get for wild type, right? It just does its thing. Sorry, I can't pull this forward because I can't. You just have to watch it do its thing. Oh yeah, you can. Good. Thank you. So move a bit forward. Okay, thanks. Then you get to the barrier, and now you would be putting them on the auxin. Now it doesn't help the guys that are already, hyperbolically or exponentially failing because they're already going down the drain. They're out of the well. There's no well. Sorry, they're just falling down the side. But those guys down here don't even know this happened, right? They're still in the stochastic drift part of the trajectory anyway. They're under the barrier even for the wild type, so they don't know that you just did anything to the upper side of the boundary. So they look exactly the same. And can you just run it to the end? So when you do this to the end, you then get this pink guy here who will just go and go and go and go and eventually drift to the new barrier and get destroyed at day 50, which is the wild type for the DAF2. So this is exactly what's happening. So to illustrate what this means, so this is what the data looks like experimentally, and if you do this in the simulation, that's what it looks like. So it's exactly reproducing the sort of effect that some of those look like the treated one. There's some effect to do with the distributed broadening where there's some detriment if you do it somewhat early, but then late, that doesn't matter anymore. So, and you don't need to resort to rejuvenation or removal of damage or something paradigm because it just comes out of the drift. And so just to make the point here, these guys here and these guys here are completely indistinguishable to the system, right? They're below, on this one-dimensional aging model, they are completely the same. They're drifting somewhere between 0 and 10 on a stochastic trajectory, and they could be doing this for 5 days, 20 days, or 2,000 days in a large enough combo. It doesn't matter. As long as they're still there, none of where the barriers are matter to them. So I thought that was a neat experiment to say, well, can we actually explain some really surprising data that lots of people have been looking at using a very simple toy model that looks at the process of the non-aging animals without all the potential and without the stochastic signatures. And of course, you can get other stuff out of it. You can get the shape of the lifespan curves. You can calculate a lot of things, which we are actually doing, and we're actually taking some of the strains from Colin to do some of those experiments. And of course, we're going to measure damage. But I think it's neat if you're a physicist and you come across the chance to doing a test of a toy model, then you should do that because it can be illuminating. And so I thank Peter and Kirill who helped with this, and also Fengshen and Brian because they're involved in everything we do. And I will take some questions.
So I love those simulations. I'm thinking back about the Smurf talk, where the number of Smurfs goes up kind of linearly with age. And if we can imagine that, would you imagine that that would be crossing a threshold into an exponential and irreversible kind of one? And so do you get that the number, if you look at the number of simulations crossing the threshold as a function of time in that diffusion phase, does it go up linearly with time, or?
So I mean, the drift itself is linear with the amount of, that's where the temperature comes in, right? Yeah. The amount of drift that reaches the first barrier is linear.
Linear in time.
Well, okay, so the radius is linear in time. The fraction of it is complicated because initially it goes up, okay, so look at the bell curve, right? Initially nothing reaches the barrier, right. So you're zero. And then it goes up in a sort of way that you could imagine is exponentially, although technically it isn't. It's rapid because right. You're pushing the edge of the Gaussian. And
it's what's called the inverse Gaussian binomial distribution.
Exactly. And that's why you get an Earth function for the survival, right?
And that's maybe
and then when you get to half and you get to modal, essentially it has to plateau because half the population has gone through, and eventually it will decline if you
Great.
Thank you, really nice talk. I was wondering, is the effect of the intervention reversible? Like if you remove the drug, would you then suddenly lower the threshold and then you'd get a big die-off as they suddenly become exponential?
Yeah, so that's actually would be a cool experiment to test this, right? I don't actually know whether you can, see that's why college should be, whether you can actually withdraw this and they revert back to wild type. But that would be exactly, if you suddenly, and then they would all go hyperbolically and die on the spot, that might be interesting, yeah.
Thank you.
Great talk, Jan. I really enjoyed. So my question is, so you have this nonlinear equation where you're showing that in some conditions it's stable, other conditions unstable, right? So you're moving from an attractor state to another attractor state, I guess. Which is
well, I mean, the unstable one is there's no attract. I mean, there might be attractor downstream, yes, right, but basically that's a disease, right? It's some sort of, I mean, like as Peter said, any exponential change in physiology is not going to be good.
State transition, for example. Yeah. Okay, interesting. I just want to know a simple question. What is the next step? You know, at the molecular level, are you able to find the genes that are responsible for the state transition?
Yeah, I mean, so the first thing is, I mean, okay, there are several answers to this. We have this paper where we're essentially trying to look at the noise and the damage accumulation, right? You're looking for two things. You're looking at the amount of intrinsic fluctuations in the system, and you find a measure of that. And you're looking at the amount of loss of potential or damage accumulation, the amount of stochastic signal. And of course, you can think of possibility of finding genes that would affect either of those two traits. Like you can do this with comparative biology, or you could do this, by doing experiments to map that. But the first thing I'm interested in is actually, and Peter also alluded to this a little bit, is actually sort of biomarkers, clocks, classifiers, right? Because you can measure some of those things. So for instance, and again, we're not talking about worms, right? We're talking about the stable case. If you get a difference in the autocorrelation function in the amount of, 1 minute left, in the amount of similarity between you and yourself at a later time, and you can get that from medical records or tracker data or repeated measurements, then you have a measure of how far an individual is down that trajectory, right, of how close to failure they are. And again, that's what you do for ecosystems. You look at, critical slowing down to detect this. And I think for me, the next step is to develop biomarkers and clocks that might measure this. And then I know, of course, Gero is a drug company, then, they're interested in identifying targets. But I think having the framework really helps you think about how to analyze omics data, let's say it this way. That was the next step.
Very interesting, thank you.
Time's up. You can ask me anytime.
I'll ask you later.